third type of materials that we can use for the fabrication of tissue engineering scaffolds are going to be synthetic polymers. The advantages of using these synthetic materials is that they have controlled properties and degradation kinetics. We have control over the composition and the molecular weight of these polymers, of course. They are also easier to process than some of the naturally derived materials. The synthetic polymers are listed here. A lot of them are the ones that we have already mentioned for other applications like drug delivery or biodegradable polymers. We have things like polyethylene glycol, which as we know is a very water-soluble hydrophilic polymer that it's very suitable for the preparation of hydrogels. We then have some of the polyesters like polylactic acid, polyglycolic acid, copolymers of polylactic coglycolic acid, also polycaprolactone, and some polyanhydrides that are going to be utilized mostly to provide biodegradability to the material. But remember that these are going to be hydrophobic, water-insoluble polymers, and so typically they are not going to be making the bulk of the material because the tissue needs to be very hydrophilic, and so they would be utilized in conjunction with other polymers such as polyethylene glycol. They would basically be utilized as linkers that would be degradable. We can also utilize polyacrylates and polymethacrylates, such as those that are listed here, which include polymethyl methacrylate, polyhema, polyacrylic acid, and methacrylic acid. And then finally, we can utilize other polymers such as biodegradable polyurethane elastomers and amino acid-based polymers, or basically polypeptides that are synthetically designed or synthetically made. In terms of the fabrication methods that are utilized for the preparation of these tissue engineering scaffolds, the methods are going to be such that they're going to lead to the formation of interconnected pores. So because the system needs to be a porous network that will enable cells to live that has enough porosity for cells to be able to be within and be able to have good oxygenation, good transport of nutrients and waste. So the methods include particulate leaching, which is shown here on the top right. Basically, in particulate leaching, what you would use would be particles typically of a salt, for example here sodium chloride, that would be grinded into small particles of different sizes that you might want to utilize. You then would apply those small crystals of salts together with a solution of the polymer in dichloromethane. In this case, they're showing polylactic glycolic acid. So the polymer is not soluble, or actually I should say, the salts are not soluble in the solvent that the polymer is soluble on. This is dichloromethane, but it could have as well been, ethyl acetate or even acetone, for example. So as long as the solvent, the polymer is soluble in that solvent, but the salt is not. So what you get here is a heterogeneous mixture of salt crystals surrounded by the polymer solution. We can then remove the organic solvent, leaving basically a solid polymer that is surrounding those crystals. And then we can submerge that solid polymer composite into water, where now the salts are going to be extracted because the salt is soluble in water while the polymer is not. And so at the end, what you're left with is the porous PLGA sheet. So that's particulate leaching, where we're using the salt to create the pores and that salt is later removed. Freeze drying is another method. This is also called lyophilization, and it's basically a process where we would create a solution of our polymer in a solvent at a low concentration. We would then freeze that solution and then subject it to lyophilization, which is basically removing the solid solvent directly from the solid to the vapor phase. So lyophilization, that phase conversion. What you're left with is basically the network of the polymer as it used to be whenever it was in solution. You basically get something like a cotton candy configuration, if you would. So it's a network. The next method is phase separation, where you would basically utilize a non-solvent to be able to generate kind of droplets within your polymer, and then you could remove that non-solvent to leave pores behind. The next method is electrospinning, which is shown here on the right. What you do is that you begin with a polymer solution. That polymer solution is applied through a spinneret that is to which we apply a high voltage. That basically results in, as the polymer is injected or ejected from the syringe, that voltage drives it to go toward a collector that is grounded. And in the process, because we have very fine or very narrow tube of polymer solution that is coming out, that undergoes very high transport, and basically the solvent is able to be evaporated very quickly. And so once the polymer actually reaches here, what you have is solid polymer fibers. And of course, the solid polymer fibers continue forming and continue just depositing onto that collector, and you end up with a mesh network of the solid polymer. Three-dimensional printing or 3D printing has also been utilized to generate these porous structures, because of course you have high control over the geometry that you are able to form. Lithography is another method where you can create very complex architectures. Lithography can include both photolithography as well as electron beam lithography. And in both methods, basically, you're going to be or at least in photolithography, you're using a photomask that allows you to preferentially irradiate certain areas of your pre-polymer solution, leading to cross-linking or curing of the polymer in those locations, and thereby leaving you with a geometry that is the opposite of what the photomask basically had. The last method is hydrogel synthesis, and in hydrogel synthesis, basically what we're talking about is polymerizing monomers through free radical polymerization to generate a network structure, as long as we also include multifunctional monomers. We could also utilize click chemistry or ionic gelation or affinity interactions to generate chemical or physical hydrogels. Now, we have already mentioned that hydrogels have very good properties for tissue engineering applications, because their mechanical properties basically resemble those of soft tissue. They are highly porous, which allows for good transport for nutrients to and from nutrients and metabolites out of the cells, so that makes them very suitable for this application. They're also very easily synthesized through a number of processes, which we already have discussed in previous lectures, both chemical and physical, and they're easy to functionalize to provide bioactivity, because remember that we have to provide cell adhesion peptides, growth factors, and the right mechanical cues to enable the cells to grow. Now, so the following biological factors need to be included within synthetic scaffolds to enable tissue engineering development. Basically, as we said before, we need to be able to provide properties that are very similar to those of the extracellular matrix. That includes the presentation of certain biological factors, such as cell adhesion peptides, growth factors, hormones, cytokines, nucleic acids sometimes, and it's also important not just to present them, but to present them ideally in the proper spatiotemporal coordination, or basically in the proper sequence. And so there's a lot of research into finding out, what is that proper sequence? Is it actually important or not? Does the spatial presentation also play a role or not? There's a lot of groups that are working on that, looking individually for each of those factors and how do they affect the regeneration of different types of tissues. Now, degradation capabilities is also important. Basically, we know that we want the external material to degrade over time to lead way to the tissue regeneration. And because of that, the degradation kinetics must match the rate of tissue formation. We said that if the degradation is too quick, then you basically lead to a collapse of the whole biomaterial because there will be no support for the growing cells to continue on. On the other hand, if the degradation is too slow, then you're going to be impeding cell growth and tissue remodeling, and that's also a bad thing. Now we can, in order to provide that degradation capability, we can either use degradable materials as a bulk for the preparation of the whole hydrogel, or not hydrogel, yeah, for the whole scaffold, or we can utilize degradable linkers. Degradable linkers is what would typically be utilized for the generation of hydrogels, because we would need to utilize hydrophilic polymers that are linked through these degradable linkers. And then in terms of bulk materials, what you could do would be to utilize a scaffold such as those made through salt leaching or electrospinning that might just be made from one of the polyesters and that don't need to be very hydrophilic, but that would be suitable for certain applications. The last thing is mechanical factors, and the mechanical factors include surface topography as well as the bulk mechanical properties of the material. And it's been shown in many papers that differences in these properties can actually regulate cell orientation, cell differentiation, and cell function. We're mainly going to be showing right now a couple of slides on how to incorporate cell adhesion peptides, how to incorporate growth factors, how to incorporate degradable linkers within hydrogels, and then at the end give some examples of some research where they're utilizing either some of the mechanical factors or some of the growth factors to induce specific functions in the tissue engineering scaffolds. All right, so here on the bottom left, what I'm showing is a hydrogel that is trying to be developed in such a way that it presents both cell adhesion peptides as well as cleavable linkages and growth factor presentation. And in this particular slide, we're focusing on how to incorporate those cell adhesion peptides. Remember that the cell adhesion peptides are derived from extracellular proteins, specifically fibronectin and laminin. And we mentioned already several times the fact that the RGD peptide has been identified as critical for cell adhesion through integrins. In addition to that, we have certain peptide sequences from collagen that have been identified as suitable to enable cell adhesion, and then certain other sequences from elastin that are shown here. These letters represent amino acid sequences, and the table on the right hand side is basically showing a number of different cell adhesion peptides, their origin, whether that's fibronectin, laminin, collagen, or elastin, and then the cell receptor that those peptides are known to be bound by the integrins. And then on the right hand side is showing what types of monomers have been utilized to be able to incorporate those peptides into hydrogel networks. The next slide actually shows you one example. So what we have here is that we're trying to incorporate the RGD peptide motif into a polyethylene glycol hydrogel. On the right-hand side, you can actually see the structure of the peptide that was incorporated. Notice that they're using a cyclic RGD sequence right here. This is arginine, glycine, and aspartic acid, or RGD. And so the main reason why they're using the cyclic structure is that the cyclic structure of RGD has been shown to actually bind with much higher affinity than the linear sequence, about 240 times, in fact, higher activity than the linear RGD analogs. And the reason for that is that in the real peptide, in fibronectin, the RGD sequence is actually presented at the tip of a loop, and it has a spatial constraint that has been found to be to increase the affinity. And so here what they're trying to do is to replicate that kind of loop synthetically to give it better affinity than the linear sequence. In the loop, they also include phenylalanine here, and then they provided a tail to enable us to attach that peptide to the rest of the polymer. And just to notice on this right-hand side, we have two lysine amino acids, each of which provides an amine group, a primary amine group, to enable us to do chemistry that can enable it to be attached. So what they did is that they took this peptide that has those two amine groups, reacted it with an acrylate, polyethylene glycol spacer, and N-hydroxysuccinimide, which is this, which is reactive toward primary amines. So this part is going to attach to the amines here, and that's going to lead us to forming this kind of system where this represents this whole cyclic RGD. This was the linkage that included the amine groups, and then we're going to have actually I should say this is the linkage with the amine groups, and we're going to have one of those acrylates on one side and another of those acrylate pegs on the other side. Now by having this is a pre-polymer that we can further polymerize by taking advantage of the acrylate groups to be able to include this as part of a growing polymer network, which is our hydrogel. And so that's exactly what they did. They actually took the cell adhesion peptides that have already been functionalized with the polyethylene glycol and hydroxysuccinimide, which is basically this, and then they polymerized that together with polyethylene glycol dimethacrylate to lead to the formation of hydrogels. And on the bottom left, what you can see is the actual data, where here they have a hydrogel that is prepared in the absence of RGD peptide. And on the right-hand side is basically an identical hydrogel, but that includes the RGD peptide. And both images is after the hydrogels have been seeded with cells and incubated for 6 hours. And as you can see, basically after the 6 hours, no cells have adhered to the polyethylene glycol hydrogel that lacks the RGD peptide. But on the other hand, the one on the right is full of cells. So the RGD peptide is absolutely necessary for cells to be able to attach to this kind of hydrogel. All right, the second thing that we want to include in those hydrogels, again, is degradable linkages. And for the case of hydrogels, really we're talking about including linkages that can enable the hydrogel to be broken. We already mentioned that we can utilize hydrolytically cleavable linkages, such as polymers that are polyesters or polyanhydrides, but those tend to be hydrophobic. So a lot of times, instead of using those, what we could do would be to utilize polypeptides that can be enzymatically cleavable. And of course, we can design those peptides based on sequences that have been already identified from other proteins as being cleavable by enzymes associated with the cells of the tissues that we're trying to develop. So here are some of the enzyme-sensitive peptides that have been identified from different origins and that are known to be cleavable. And in each of these sequences, which are amino acid sequences, there's an arrow. That arrow indicates the location where that peptide is known to be cleaved. So basically what we would do would be to use this as basically a chemical linker, and we would attach it on either end to the rest of the polymer structure, and then knowing that it would be broken wherever the arrow is. The origin of those peptides are shown here on the second column. The enzyme that cleaves them is listed here. MMP stands for matrix metalloproteinases, which are enzymes that are typically utilized to cleave the extracellular matrix. We also have plasmin here. Plasmin is the enzyme that is responsible for cleaving fibrinogen or cleaving fibrin clots, I should say. And then here we have elastase, which is another enzyme associated with the cleaving of elastin peptides, and then another matrix metalloproteinase. And then on this column is showing what type of monomer was utilized to functionalize these peptides to enable inclusion of those peptides into the polymer network. The references are here, and of course this paper is from Biomaterials 2010. All right, the third thing that we need to include is going to be the growth factors that are going to make this hydrogel to be a bioactive scaffold. These growth factors are basically proteins that transmit signals to modulate cellular activities. they typically need to bind to the extracellular matrix because they're not very stable. They can basically be metabolized very quickly in the circulation. So typically we want them to be protected. These growth factors typically act by binding to specific receptors on the surface of the cells, which means that not only do we need to embed them within our hydrogel systems, but we also need to have them present and available for the cells to be able to bind to them. What that means is that basically these growth factors, if we can either embed them in a soluble form within the hydrogel structure, or we can attach them chemically to the hydrogel, but in such a way that they are connected through flexible linkers, so that the protein itself is able to attach now to a cell through a protein-protein interaction. So a lot of times also what it's done is that they might be chemically conjugated or chemically attached to the hydrogel, but through degradable linkages, such that they can be, those linkages can be cleaved, enabling the growth factor to be released within the hydrogel still, but released to be able to interact with the surface of the cancer cell or the surface of the cells of interest. Some of the growth factors that are utilized are listed here: vascular endothelial growth factor or VEGF, as well as basic fibroblast growth factor. These are typically utilized together with the platelet-derived growth factor for induction of angiogenesis, which is the development of blood vessels, which is critical for tissue regeneration. Then we have here epidermal growth factor and the transforming growth factor, which are key for inducing cell proliferation and differentiation. And then finally, we have bone morphogenic protein, which is critical for bone formation. Here in this table is also again showing different types of biomolecules, meaning the growth factors that have been included in hydrogels, and what type of chemistry was utilized in order to incorporate them into polyethylene glycol hydrogels. As you can see here, the growth factors are not typically put within the backbone of the polymer, but as linkages, as I mentioned, to make them more available to the cells, because they need to be able to bind to cell surface receptors. All right, some of the challenges to tissue engineering is, well, first of all, that it's actually really hard to be able to put all of the cells that are needed and integrate them with the surrounding tissues and have them actually be appropriately vascularized, meaning that not only do you have to generate the right material and put the right cells and ensure that those cells are viable, but eventually we need blood vessels to be able to continue supplying nutrients to these tissues, and that's something that has been very difficult to achieve. A second thing is scale up. Of course, we might be able to do this in the laboratory, but basically making one scaffold at a time. But in terms of being able to do this in a commercial scale, this is difficult. There's also a lot of variability in terms of patient response, a lot of variability in terms of the cell sources, our ability to differentiate stem cells, for example. And there's a lot of variability in the manufacturing of these scaffolds as well. There's also difficulties with the storage, the distribution, and the shipping logistics of such materials. And there's a lot of regulatory issues and ethical concerns with the use of some of the embryonic stem cells, of course. All right, I mentioned the vascularization is one of the key challenges for tissue engineering. So cells, any cell, needs to be within about 200 micrometers from the nearest blood vessel to be able to obtain enough oxygen and nutrients and be able to get rid of waste sufficiently. So that creates a huge challenge because we cannot create a large or a whole organ if we cannot induce blood flow to it, right? So the scaffold itself needs to be designed in such a way that it will either encourage vascularization, or that we might already develop the material itself with vascularization or pre-vascularize it, basically. here what it's saying is that some tissues might be okay. We might not actually need to worry about vascularization. And that is something like skin or cartilage, which require about 2 cell layers. So that's thin enough to be able to, for those cells to be able to access blood. On the other hand, pretty much any other tissue needs to either be prevascularized or it needs to deliver angiogenic growth factors in order to encourage blood vessel formation. The next few slides come actually from a review paper, and they're going to show you some of the methods that have been tried in order to try to develop vascularization of tissue engineering scaffolds. I highly encourage you guys to look at this review. It is Trends in Biotechnology, and I have put it in the track site. So take a look at that, but the actual source of this particular paper is from PNAS. And basically what is shown here is that they basically generated a hydrogel, which has, it is a collagen hydrogel, I should say, that has been, I guess, modified to be able to include channels. So everywhere where you see this red, that's basically a channel. And that those channels were then seeded with vascular cells. This resulted basically in a perfusible endothelial lined network, which is what I'm showing here. So again, we have basically channels, and those channels were then perfused with vascular cells, which is the endothelial cells. And so now we have basically tubes that are formed, are surrounded by those cells. Then depending on the culture conditions and the presence of the vascular smooth muscle cells, the structures were actually found to start sprouting into the hydrogels. And you can see that here. You can see a little bit of cells coming out from the original seeding into the rest of the hydrogel. So the black represents actually kind of solid, if you would, hydrogel, not a tube. And you can see that the cells are starting to sprout. That sprouting is basically the development of a blood vessel now, because these are the endothelial cells that line the blood vessel. And they actually adapted their barrier function as illustrated here. So here what they did is that they perfused that same kind of scaffold that still had the cells with a dextran solution that was fluorescently labeled with fluorescent isothiocyanate, and that gives us green fluorescence. As you can see, we actually have kind of some of the dye coming out from the tubes into the solid hydrogel, meaning that the the barrier has been compromised in some of the areas where remodeling might be taking place. And here on the right hand side, it was just showing some of the like the actual tube. Like if you were looking at this particular corner here, you can actually see the tube. The red indicates labeling of the cell surface, and then the blue represents the nuclei. And this right here is actually showing the cross section if you were to look at this from the bottom, and here on the right hand side is the cross section if you were to look at it from the right hand side toward the left. Here is basically a three-dimensional reconstruction of what is seen in this corner right here. This is another example where on the top channel, this channel was actually microfabricated in a collagen hydrogel. The top channel was seeded with human umbilical endothelial cells, and the lower channel was perfused with vascular endothelial growth factor. What they did in this particular project was to look at what effect would the distance between the two and the geometry actually cause on the sprouting of these cells, or the invasion of these cells to migrate toward the vascular endothelial growth factor, which, as I mentioned, is responsible for inducing angiogenesis, or the development of new blood vessels. And so it was interesting that, again, they're investigating the effect of the distance, which also means basically gradients in concentration between the source of that growth factor and where the cells actually are at, and the geometry, to see what would actually induce better sprouting of the cells toward that molecular cube. In this other paper, they were actually looking at mechanical signals. So what they did is that they have cells at the center that are embedded within different types of hydrogels of different mechanical properties. Sorry. So they have different compressive modulus specifically. And what they found is that the higher that number was, the higher the compressive modulus, the more sprouting they actually saw for the hydrogels. The last one is actually looking at the last one is looking at the effect of flow in the organization of the cells within a tissue engineering scaffold. So in this case, what they did is that they developed a perfusible network in a microfluidic device, and here they were just testing to make sure that it was perfusible, and they did that by basically putting fluorescent microbeads and watching them flow within the channels. And the idea was to then seed cells within those channels and be able to see how do they organize in the presence or in the absence of flow. And here, and I should say, in here on the right-hand side, they actually were again testing the perfusibility, in this case with fluorescein isothiocyanate dextran, so it's a fluorescently labeled dextran. And again, you can see the green fluorescence in this case represents that this dye is actually flowing well within the channels. So then they tested how do the cells actually grow within those channels, either in the absence or in the presence of flow. And what they found is that the endothelial cells actually responded to the fluid flow with their cytoskeleton organizing around that flow, basically, versus this system where in the absence of flow, the cells don't seem to align in the same way. So that was really good, that basically the presence of flow enables the cells or provides a cue to the cells to be able to organize properly in the way that they would have organized in a blood vessel. Then they tested the nitric oxide production, and that's what's being shown here. In the absence of flow, there's very little nitric oxide production, while in the presence of flow, you actually do see nitric oxide production, which is what's quantified here at the bottom. And that's just showing that not only does the organization of the cells change, but also their metabolic activity and their phenotype changes in the presence of flow. So in general, just to kind of wrap up the tissue engineering chapter, what we need to do is to be able to create a material that can provide the right mechanical cues, the right biochemical cues, and the ability to degrade to be able to enable the cells to organize, differentiate, and grow in order to provide the function of the organ that we are trying to replace. This is a very complicated problem, and there's still a lot of research and development to be done to be able to successfully achieve this, but it is a very exciting area to be in.